Hello everybody! Welcome back to Creation Myths. Just give me a sec here, pull everything up on YouTube, make sure it all looks good. Okay, I see that we're rolling. Just waiting for YouTube to make sure I can see me and hear me, and then we'll be good to go. But just waiting. Oh, now it is. There we go. Looks good. Sounds good. Okay, chat, welcome. Please remember to tell me if there are any audio or video issues as we're going. I don't need those because it's just me tonight. So let's start that over. Hi, everyone. Welcome back to Creation Myths. So tonight we are going to talk about a really interesting new paper. I have been kind of really excited to dig into this one since the paper came out. Dr. Joel Duff did a fantastic write-up of this. I link that down below along with the paper, so check that out. He really dug into some of the details here, and I am actually coming into this cold tonight. I haven't had time to go through this paper, highlight, annotate, so I'm looking at this paper having read Joel's article and what I've seen about it, just kind of through other people talking about it, but I haven't actually had time to go through this paper. So this is going to be really cool because we get to go through this paper together. So chat, what I'm going to do is put paper up on screen. You all can read it. It'll be nice and big. And if you have questions about anything that's on screen, if there's anything you read that I miss, tell me, and we can kind of do this in a collaborative way as we're looking at this paper. So let me pull it up, and then we'll talk about what we're going to do here. Let's see, make sure everything's good. Okay, everything's looking good. All right, so here is the paper. It is called Limitations of Serial Cloning in Animals, published in Nature Communications just this year, just a couple months ago. Wakayama et al. And this is a really interesting paper for a couple of reasons. First, it's just really good science. As you'll see as we go through this, it's a long experiment involving many generations. Here we go. This is a live stream. Here we are. We are actually live, not premiering tonight. It's the end of the semester, so I've been really busy getting the tail end of the semester squared away and also writing a final exam. So yeah, I haven't recorded for this week, so we're doing a live stream, which is great. We get to talk about this really interesting paper. So why is this paper super cool? First, it's just the genetics of this study are super cool. We're talking over 50 generations of cloning in mammals, and I'm old enough to remember when one generation of cloning in mammals was literally worldwide headline news with Dolly the sheep, right? So getting a write-up of a paper that goes, you can see it right there in the abstract, out to 58 generations of cloning, that's really cool. And just how they did that and kind of what they observed over the course of that in terms of the success rate, the growth rate, and all that stuff is really cool. But then on top of that, this paper has really important implications for the evolution creationism stuff, specifically genetic entropy, because I am putting this in my list of papers that disprove genetic entropy. And we will see why that is as we go through. But the researchers here, the team, did a really interesting thing at the end with that last generation that was non-viable. They did a really interesting thing that turned out, recovering some of their fitness, that if genetic entropy is true, shouldn't be possible. So we're going to go through this. We're going to do the abstract here, and then we'll go through. And again, I'm actually going through this text for the first time right now. So there's going to be stuff I miss. This is, I want to be clear about this. This is not specifically my field. So when it comes to the specifics of the techniques that they're doing, I am not an expert in those specific techniques. So there's going to be stuff here that is over my head, and that's okay. Part of the fun aspect of this is reading things that you are not specifically an expert in and kind of working through them. And that's a hard thing to do, and it's also really fun. So we're gonna do it together here tonight. So here's what this abstract says. It says mammals can now be cloned artificially, but it remains unknown whether they can also maintain their species through cloning, right? So we could do it a little bit, but can you do it indefinitely? Herein, we continued serial cloning for 20 years from a single donor mouse. Recloned mice appeared normal lifespans. Structural and lethal mutations accumulated in their DNA with each generation, as we would predict, right? The birth rate began to decline, starting with the 27th generation, and the 58th generation was the last, and they will explain that in more detail. When recloned mice from near the final generation were mated with males, their oocytes would be fertilized. Most embryos degenerated. However, a few embryos were normalized by meiosis and fertilization and developed to full term, suggesting that mammals rely on sexual rather than asexual reproduction to eliminate genetic anomalies caused by clonal reproduction. So that was kind of a lot there. The big idea is that what we saw in the cloning, which essentially we're doing asexual reproduction, is mutation accumulation, both point mutations and structural mutations, correlated with a decline in lifespan until the 58th generation was just couldn't reproduce at all. But if you allow them to recombine, you do meiosis, and then mate them, now you're good. At least better, right? You can recover some of that fitness cost. And the implications of that are that sexual reproduction, with all the inherent energy costs, the danger of it, right? The extra steps, the time, right? The on average slower rate, all else being equal, what this paper points to is that the benefits that you get in terms of generating the variation, and specifically in terms of removing harmful mutations via recombination, allowing you to select for the genomes or genotypes that have fewer harmful mutations, that benefit is sufficient to override the cost inherent to sexual reproduction, which is like a really cool thing, right? So let's see what we talk about here, all right? So we're not going to read the whole rest of the paper word for word, I promise. We're just going to kind of go paragraph by paragraph and skim and get the idea. So asexual reproduction, many species, yeah, right, potatoes. Somebody in chat was just talking about this, right? Somebody just said cloning is super common in plants. Yeah, right? So there you go, right away. Right, and we know this is the thing in plants. Different deal there. Animals, yep, a few species. Planaria can do this. Mammals cannot reproduce clonally. Yeah. And the reason for this remains unknown. I think that might be overstating the case a little bit. I mean, there's been quite a lot of research into kind of the cost benefit of sexual reproduction. And I mean, my understanding coming into this is you recombining allows you to combine good mutations, right? Beneficial mutations, so you don't have clonal interference. You don't have competition between different lineages in the same population with different beneficial mutations. And then the other thing, it allows you to decouple the good from the bad, so you can select for the good and get rid of the bad, right? So in a theoretical sense, I think that was actually pretty well understood, but experimentally, this is an extremely robust demonstration of that principle. So I think that's kind of where we're at here. So unknown, it might be a little much. Until recently, mammalian cloning was not possible. Twins, yeah. Okay, so we got some history of cloning here. Yeah. Yeah, you could use domesticated animals. Practical uses, right, low success rate. Okay. Yeah, this is a big one, the resetting to the epigenetic state, right? One of the things that happens when you have fertilization and embryonic development, you kind of reset the epigenetic profile of that structure. Because there are genes you inherit from each parent that are going to have parent-specific epigenetic profiles, and that gets revised, right? And you kind of start fresh. And with cloning, you don't do that, and that's an important step, right, for development and things. So that is definitely something really important that you have to account for when you're doing multiple kind of generations of this. Oh, and this is a really good point from Templar Warden. This work has been ongoing for 20 years, so how understood has it been at the start? I'm assuming that's start, but I've got something on my screen covering it also in StreamYard, but assuming, yeah, start. Yeah, that's a good point. Although, yeah, I think when they started it, obviously all this was less well understood than it is now. I still think saying unknown there is a little much. Okay, so DNA abnormalities do not occur and epigenetic errors don't accumulate. Mammals should be able to do it. Basically, yeah, if you don't have mutation accumulation and messed up epigenetics, then it should work. Yes, good. So we can improve cloning success by using epigenetic modification reagents. Yep. And then you get 25 generations. Success rate improved with each generation. And they initially thought that it could continue indefinitely. After continuing for 28 years, 58 generations was as good as you can get. Okay. So we aim to investigate what occurred and to clarify why mammals maintain sexual rather than asexual reproduction. Okay, so that's the setup. That's a really nice, crisp introduction. I didn't realize that was it, and I think that was really great. There is a form of science writing where you have basically what's like a 3-part introduction, where you say, here's the question, here's what's been done before and why it's not good enough or still unanswered, and then here's what we did that answers it, right? Boom, boom, boom. And this kind of holds to that form, and I really like that. You don't need these mile-long introductions where it's a lit review of the entire field. Like, give me the background I need to understand your paper, and let's do it, right? So I really like that. I like that form of science writing. Way to go, authors. Okay. So now let's get the results. So they started this back in 2005. And we increased up to 15%, 15.5% by the 26th generation. We can see that here. Figure 1, right? That's, yeah, success rate of serial cloning, generation 1 to generation 58. Peaked right up here at, like, what is it, like 26? Yeah, 26th generation. No difference in the 1st and 25th generation clones. Great. Therefore, we believe that serial cloning they thought they could just keep going. Reasonable conclusion if you're at this point in the timeline that, hey, look at that trajectory, we can just keep going. And then what happened? The opposite of that happened. So no luck there. We get joining late, starting at the beginning because there is a new Nova doc about Artemis 2. This, I'm assuming you mean this takes precedence though, and I appreciate that. Thanks for being here. So, okay. 3 to 4 generations each year, decreased after the 26th, 57th round. All 58th generation recloned mice died the next day. So no viable mice in the 58th generation. That's the key there. The original donor mouse was a female, and therefore all cloned mice were female, same coat color, whole genome sequencing. Also confirmed these clones originated from the donor mouse. Okay, cool. So this next section, placenta and lifespan of cloned mice, abnormal placentas arising from epigenetic errors. Okay, so basically what that means is that it wasn't mutations to the gene, any genes that caused the problems with the placentas, or at least were not necessarily associated with them. It was differences in gene expression patterns during early embryonic development, so the placenta didn't form correctly, and that's mediated through differential expression of genes. And there were probably mutations in those genes too, but apparently the problem was the expression pattern, which is what epigenetics is gonna regulate. Yeah. Also, just FYI for everybody. Just keeping an eye on the chat here. Good to see everybody here. I appreciate it. I see, what do we got, like 50 people here? Heck yeah. Let's talk about cloned mice. Let's do it. People hanging out to talk about cloned mice. Love to see it. Yeah, so we're dealing with body weight here. Looks like My guess is body weight went down, increased over successive generations. Okay. Oh, their body weight nor placental weight increased. So the mutation slightly heavier, but similar to the control lines. Mean placenta weight. Okay, so not too much is going on there. These placentas are significantly different. Also examine the average lifespan. The average was about 2 years, similar to that of control mice. Okay, so not too much there. Okay, so here we have this, what is this? Trichostatin A, which is a known enhancer of nuclear reprogramming. Basically, it's going to induce the epigenetic reprofiling that occurs early in embryonic development. So if you can increase that, then you should increase the success rate of your cloning. That's cool. So the success rate for G51 clones significantly lower than with TSA treatment. Even more than 50 transfer cytosomatic cells, recloned mice retain sensitivity to TSA. Okay, so the TSA did its thing throughout the experiment. Okay, cool. Let's get to the mutations. That's the part that I want to hear about. I want to hear about mutation accumulation. So hang on 1 second right here. I want to highlight a question that we got in chat here from Joseph Grasser 3746. If the scientists interbred the recloned mice, would the offspring benefit from the sexual mechanism? I want everyone to put a pin in that because they did that later in the experiment. So we're going to see if those really low fitness clones from later on can benefit from a little bit of recombination, right? Spoiler, the answer is yes, which is really, really cool. So we're gonna see exactly how that works. Epigenetic abnormalities. Okay, so there's a little weirdness with so epigenetics is chemical modification of the DNA, usually with methyl groups, but you could also methylate or acetylate the histone proteins that associate with the DNA. And by methylating or acetylating, you're adjusting the level of like the density of the DNA packaging. The denser it is, the lower the expression of that region, because you can't get like transcription factors and polymerases in there to affect the genes if it's densely packaged. So in general, heavily methylated DNA, lower expression. And what we're seeing across these cloned lines is that that methylation pattern, that epigenetic regulation is all thrown off because part of every generation you kind of reset that a little bit. And if that's not happening, then that's going to cause problems. So I don't know what specifically these genes are, but I see we got trimethylated histone. So that's a histone with 3 methyl groups, and it's going to condense everything. And then acetylated histones, that's acetyl groups are going to cause it to become more diffuse, right? So in figure 2, they were similar between NG1, except for, okay. Both groups are markedly different from FC zygotes. What is FC? Did I miss that? Anyone see what FC means up above? We got to find that. This is the fun part of reading it. There we go. Fertilized control. There we go. Got it. Okay. FC is fertilized control. So those are the sexual lines, right? So if you're comparing the clone lines versus the FC is the sexual ones. That is kind of the baseline for each generation for sexual reproduction. By the way, this is a lot of what reading a paper is like, where you're constantly like, wait, what does that mean again? Let me go back, see what that means. Okay, now I can go back and understand the paragraph a little bit better. Okay. Okay, so these results suggest that epigenetic abnormalities did not accumulate in cloned or recloned embryos, at least at the 1 cell stage, so which is to say in the zygote. Or, well, it's not a zygote because it's not fertilized, I guess. So I'm still thinking in terms of sexual, but it, would be the zygote there. Okay, so now we're down here. In vitro development quality of G51. So this is G51. Remember, it went until generation 58. So this is a little bit before the end, but if we just go back up here real quick, you can see that by G51, the success rate was very, very low. It was, I guess, there. So you're looking at about 5%. It did jump up to almost 8, but then it crashed back down. So we're kind of at the end here. So we've got in vitro until they reach the blastocyst stage, which is a few like 100 cells. It's where you have a trophoblast. It's like a single layer, looks kind of like a soccer ball. And then inside that you have what's called the inner cell mass. And it's the inner cell mass is what's going to become the embryo after implantation. The trophoblast is going to kind of form these projections into the endometrium to like acquire nutrients. And then the embryo itself is going to develop from that inner cell mass. That's what's going to undergo gastrulation, ultimately. Okay, so in figure 2D, let's go to figure 2D. Right here, we have development rate, and it says 2-cell, 8-cell morula, blastula, what is XB? What is EXPB? There's a lot of expansion, experiment, explanation. What is oh, there we go. Expanded blastocyst. Okay, there we go. Should have figured that one out. Expanded blastocyst. So what you can see here is that you have, what's NG1 again? That should be generation, newly generated clone. So that's our clone generation 1. And then FC is your control, and then G51 is your all the way at the end of the line clone. So the interesting thing here is if you look at the development rate, right, it doesn't actually decline that much between the beginning of the clone experiment and the end of the clone experiment, right? Those are pretty close together, but it's a lot lower than the sexually reproducing line. So there you go. That's cool. Good to see. Okay. Evaluate the embryo quality. Some blastocysts were analyzed. Cell number, inner cell mass, allocation, distribution. So the embryos looked the same. The average inner cell mass cell number was higher compared to the control embryos. Okay. RNA-seq is looking at gene expression profiles. Exhibited similar gene expression. Okay, so everything looks kind of the same there. That's going to be, is that even in a figure? Figure 3A. Oh, that's down here. Okay, so the gene expression profiles were comparable, so that's fine. A little bit surprising for me, having just read about the epigenetic stuff with the placenta, but okay, I'll take it. So now, here we go. This is where we're going to get interesting here. Production of offspring from recloned mice by sexual reproduction. Because again, what we're seeing here is mutation accumulation over the course of this experiment. Did they say how many mutations at any point? It's probably a lot. It's probably like thousands. We'll get to it. It was probably a lot. Oh, it's literally in the next paragraph. Or on the next page. Yeah, there you go. So 4,000 single nucleotide variants and small indels. Okay. So yeah, we're talking a lot of variation here. We're up here. So we're looking at serial cloning cannot continue. The recloned mice remain healthy except for G58. So could you take previous generation and then mate them with normal mice from outside the experiment to evaluate how they could reproduce? There was no, so they did this for the control mouse and then G20, which is in like the good range of the experiment. If you go back up here, G20 was a pretty, I mean, it had that dip, but it was, you know, reasonably high success rate around G20. And then 50 and 55 was in that decline period at the end. And what would be really interesting are the results with 55, right? Because they're right on the cusp of being completely non-viable. But we want to know, can we outcross them? And is that going to do something to help recover some of the cost of those mutations that have accumulated? So let's look. Okay, so no differences in age, first delivery. Okay. Litter size declined sharply. So it looks like, yeah, 10-ish versus 2 or 3. Which is not good. But the number of oocytes recovered was recorded. Although, let's see. Okay, so it's about like how many oocytes you get. This has been going, welcome. Sorry you missed the beginning. We've been going for about 25 minutes. About 25 minutes. The decrease in litter size was not due to a reduced number of oocytes. Okay, so it was actually, so one of the hypotheses was maybe they're just not ovulating as much in the clone lines, and that's leading to fewer embryos and a smaller litter size. But that's not what happened. There was the same amount of oogenesis going on, but it was just fertilization, development, implantation, something in there was not working. I really appreciate, and I hope everyone following along with me appreciates how the authors here are like kind of checking their work at each step, right? Are the lifespans the same? Are the growth rates of the embryos the same? The placenta's the same? The epigenetic profile, the gene expression, like we want to check like each one of these factors and kind of cross them off the list as potential explanations for the observations of this decline in fitness, right? So that's really cool. So far, I'm really liking how the authors are constructing this and going about trying to explain the observations here. Because remember, they started with, hey, we got this improvement, maybe we could just keep going. And if not, continue the improvement, maybe just stay flat. And then instead what happened is it dropped off. So now we're trying to explain that. And we're going through, okay, was the gene expression profile different? No. Was this different? No. When we outcrossed them, they had fewer offspring. Was that due to fewer oocytes? No. Like they went through all these different kind of questions. And that's really cool to try to, systematically work your way through and figure out what actually explains the observations. So now let's jump down here. So we're just right here. We eliminated number of oocytes as an explanation. So then the next generation, so this is really cool. This is really cool right here. So remember that G50 and G55 litter size when they outcross them was about like 2 to 3 mice on average. And then when they did it again for the G55, it increased to 7. And that's in figure 3C right here. That's really interesting. Yeah, so you can see it was about 10, 3, 2, and then it jumps up. That's really interesting. So let's look at, so the placentas from the grandchildren were smaller and nearly the same size as those from the fertilized embryos. So what this is showing here is this right here, this larger placenta, that is abnormal, right? This is closer to your typical placenta morphology, right? So the fact that just within those, you had your cloned line G55 or G50 in that case, and then you had offspring of that and then grandchildren. And the grandchildren placentas exhibited almost wild type morphology, right? Which is super cool. So you took these 50 clonal generations to build up those mutations, and then 2 generations of meiosis, and you've recovered a lot of the, I should say, you have undone a lot of the harm, right? The detrimental effects of whatever mutations, just a little bit of recombination, and you're right back in the ballgame, right? So that's a really interesting finding. And I think they mentioned above that the larger placentas was part of the explanation for the high failure rate. As you got further on, the placentas were not operating in the typical way, both morphologically and probably physiologically. So this is a really good point, Templar Warden. I got the impression the placenta size was an epigenetic thing. Right, that's what we read above, and that seemed to be what they were saying, is that it was epigenetic. We can go back up to that section, actually. So that was up here. Where was the one with the placenta size? Here it is. Here we go. So it was this paragraph up here. This is on page 2, if anyone missed it. So it's like on the right side of page 2, if you're following along. So the body and placenta weights were measured. These are the fertilized control embryos, right? So let's look at the placental weight, 2 to 3 times heavier than that, but comparable to other clones. Yeah. So the cloning caused the placental weight to get bigger, and then it stayed larger throughout the experiment. And then immediately you do a couple rounds of meiosis and it gets back. And I think that is actually good evidence that it is an epigenetic thing. Um, yeah, I'm just looking here. These placentas were significantly different compared to the control mice. It's lifespan, the average approximately to some of the control mice. Okay, so the lifespan was fine, but the placenta morphology was a big problem. And that led to saying, hey, is it an epigenetic thing? Because you have that kind of rewiring of the epigenetics very early in embryonic development prior to placental formation, and then maybe that's explaining it. I guess they didn't really resolve that one way or the other. At least it doesn't seem like it, right? But again, I'm just kind of skimming through this. I'm not. If I was giving this paper in like a seminar, like if I was doing a graduate seminar where I was presenting this paper, we wouldn't do this in an hour. It would take me a solid afternoon of working through this to make sure I really understood it, which is not what we're going to do here. We'll be here for 4 hours. So yeah, I think that was a really good question, and I'm not actually sure what the answer is. Maybe we'll get it as we continue reading. Let's see. So let's see here. Oh, we were down here now. Yeah, we were down here. Okay, let's look at the recloned mice. So that's looking at the oocytes and then the whole genome sequencing, and that's what I really want to get into. Okay. So, oocytes, near final generation, reduced quality. If mutations accumulate through serial cloning, diploidization. Ooh. Yeah, right. So what's happening here is that you have oocytes, right? Those are haploid. Well, primary oocytes are haploid. Secondary or are diploid. Secondary oocytes are haploid. So I'm not actually sure what they're referring to here, but assessment of their effects on early development. So I'm not sure which diploid or haploid they're looking at here specifically, but what can happen is if you have a haploid genome that has harmful recessive alleles, and you cross it with another haploid genome, right? Now you can have heterozygotes where it's one dominant, one recessive. You cover up the recessive, potentially disease-causing allele. You don't see that phenotype. But if you're cloning and you have a haploid cell and then it becomes diploid through like a duplication or something, well, now anything that's recessive and harmful that's present is going to be expressed because you're going to end up homozygous for that trait. So heterozygous, right? Hang on, let me go back a step here. If you're diploid, like we are, all of our somatic cells, everything except our gametes, are diploid. So that means you have two of each locus in your genome. Two of each location is what locus means. So each place in your genome. You have two of them, one from each parent. So if you are homozygous at a locus, it means those two copies from each of your parents are the same. If you're heterozygous, it means they're different from each other. So, for example, if you are homozygous for type A blood, then you got A from one parent, A from the other parent. If you're heterozygous for like AB, for example, then you got an A from one parent and a B from the other. Now, if the alleles we're talking about are something that causes disease, a recessive genetic disorder, cystic fibrosis, for example, right? If you only have one copy and it's recessive, well, maybe the one from the other parent's dominant, covers it up, you don't get that disorder. But if we're cloning, you have your one copy, and then it becomes diploid, and now you're stuck, right? Now you're homozygous recessive for a disease-causing allele. So that's what we're talking about here. So, yeah, so what this is saying is 91.3% of embryos from control mice was BDF1. Where is that? I missed that one. Hang on, let's find BDF1. So I don't know what that means now. BDF1. Is that the first place where we see it? Where's BDF1? Oh, that's just a type of, okay, it's just like a type of mouse. Okay, cool. Got it. This is me not being a mouse person, not knowing what that is. Okay, got it. That's just a type of mouse. Okay, okay, so basically 91.3% develops into blastocysts, so 12.7% of the G25s and none from G53. Okay, so basically they're saying, remember, these are all female mice, right? So females will develop oocytes during embryonic development, and then they arrest in prophase 1. So you can take those oocytes, let them finish oogenesis, go through meiosis 1, expose them to sperm, they'll complete meiosis 2, karyogamy will happen, and now you get a zygote, and we're going to see what percentage of those will develop to the blastula stage, or the blastocyst stage. And as you can see, it's 91.3 for the controls, 12.7 for the cloned mice, which is a very low number, and none by the time you get to the end of the experiment. So that's a problem. So then this is really cool. We're going to take the oocytes and fertilize them with sperm from control males. Even if the oocytes have a bunch of lethal mutations, the sperm genes could improve that. And then that number jumped. Look at that. Went from, what, 13% up to 40%, and then 0 up to 20%. So that's a huge jump by outcrossing them with the control males, right? I wouldn't call them necessarily wild type males, I guess, because they're still going to be, these weird experimental mice. But that's huge. And what I would like to know now is that due to the crossing that allows you to filter out the mutations, or is that due to the epigenetic reset we've been talking about? So which is it, right? Is it a mutation thing, or is it an epigenetics thing, or is it both? In bio, the answer is usually both, right? But that's what we want to address here, is which side of that question are we on, right? So, here we go. To determine whether the abnormalities originate from the oocyte nucleus or cytoplasm. Oh, yeah, that's another one. It could have been, so some I didn't even think of. In oocytes, these are the big, right? These are cells that are going to become the eggs, the ova. So they have a bunch of stuff going on in the cytoplasm, right? Because that's where all of the stuff in the zygote comes from. The sperm is just donating DNA, and that's it. Everything else is on the egg side of the ledger. So could there be stuff in there that's like, maybe it's unstable, it's whatever. Maybe that's the problem. So let's see. It was removed, maybe transferred, pathogenic embryos, confirming normality. Okay, so that was not the explanation, but it's good that they checked. Cool. So now let's look at the genome sequencing, right? We've got 30 times coverage, very cool. G6 to 57, supplementary table 11. It's also in figure 5, which we'll get to. Here's the sequencing stuff. But for those of you who will appreciate this joke, they also hid a bunch of the data in the supplementals. That is a joke, folks, for people watching later. They didn't hide the data in the supplementals. That's just where you put your raw data. It's a joke. 3 of the individuals, long read coverage. Okay, that's fine. So they did a bunch of sequencing. Great. Constructed the genetic lineage for each. Yeah, there you go. You all get it, right? The ice cream was hidden in the fridge. Freezer? I put my ice cream in the freezer, but you get it. We get it. All right. So the phylogeny matches no genetic contamination. That's good. Accumulated over generations with about 37,000 3,700 single nucleotide variants and 80 insertion deletions. So that's really interesting. A lot of single nucleotide variations, dozens of deletions. I do just want to pause in our doing real science for a second and call out a friend of the channel, Dr. Rob Stadler, over here. In a recent video of his that I reviewed with Erica and Dapper Dinosaur, he said something to the effect of the duplications are more common than single nucleotide substitutions. And sir, that's a lot of mutations that are not indels. Okay, like just look at the numbers. It's point mutations are the most common mutations. So back to our regularly scheduled actual science programming now. Okay, on average, each generation acquired 69.4 SNVs and 1.4 indels. The SNV mutation signature resembled that of naturally occurring germline mutations. I'm laughing. You all know why I'm laughing. Because this is an experiment that has no selection, right? You're not doing recombination. You're just going from one generation to the next. And any mutations that occur in the germline of one are going to be found in the next, right? They're all going to be passed on with no filtering. And then the mutation accumulation rate over the course of the experiment approximates the germline mutation rate. So there are implications in this experiment, not just for genetic entropy, but also for Jeanson's time to most recent common ancestor calculations, because here we actually have an example where across dozens of generations, the mutation accumulation rate actually approximates the germline mutation rate, which is the opposite of what we see when you do like multi-generation pedigrees in humans, for example. So that's a really funny one. I'm going to just bookmark that mentally, and I'm going to come back to that later in other contexts, because that, my friends, is pretty cool. Okay. So we also see a bunch of structural variants. So these are going to be like insertions and deletions. Table 2, whoa, didn't mean to click on that, sorry. Table 2 listed something, and then I clicked on it, and it went. Here we are. Table, where was I? Here it is. Table 2 lists 16 large structural variants. Ooh, 40 kb, that's 1,000 bases. Which are probably going to affect phenotype. And then, oh, here, yeah, this is from Prixie Purple. This is a good point. Really, though, what biology paper doesn't have implications for creationists? Yeah, it's like this experiment on its face has nothing to do with anything that creationists talk about, really. I mean, it's mutation accumulation and die-off, but it's the conditions are so far removed from actual populations in nature that, like, what would you extrapolate from that in terms of like genetic entropy? I mean, I'm sure creationists are going to try, but good luck. But this is a really fun example of how, like, just basic experiments that you see, you could probably go to any issue of some random genetics journal and find stuff that shows you that this or that creationist argument is wrong. And this paper's going to do it in two ways, it looks like. Okay, so we got big structural variations, and those appeared kind of in that range. Loss of an X chromosome, loss of heterozygosity, chromosome 4, translocation. Those are probably highly deleterious, I would think. I wonder if that correlated with a drop at all. Like if it was like here or there. Was that like 37? Yeah, that was in that range. So I wonder if those correlated with specific structural variations occurring. Let's see. Telomere length varied widely. Yeah, telomeres are weird. Yeah, it doesn't seem... That makes sense that wouldn't do it. Okay, here we go. Let's look at the mutations of the 4,000 something. High loss of 0.95% were classified as high loss of function or moderate. Missense is when you switch amino acids. So it was one amino acid, now it's a different one. And that might mess with the protein, or it might not. It depends. It depends on if those amino acids are biochemically similar to each other. So, like if it's a small non-polar amino acid replaced with another small non-polar amino acid, is probably not going to have much of an effect. But if it's a small non-polar amino acid replaced with a large acidic amino acid, then that's going to mess with the folding of that protein and probably have some more significant effect. All right. So let's see. Significantly higher proportion in the latter group. So you have not that many harmful mutations early on, many more harmful mutations later on, like more than twice the percentage. And that's interesting because it probably is looking at two things. You're looking at homozygosity, right? So you had to have two of the mutations or two mutations that completely break a gene or that sufficiently reduce the function of that gene to affect the phenotype. You need to have two of those in the same gene, one in each copy, right? So it's going to take time for both of those to happen. Or what could be happening there is epistasis, right? Where you have one mutation has an effect and it's pretty minor, but then another mutation occurs and in combination they have a significant harmful effect, right? Whereas either one of them alone might not have that effect. And in both cases there, homozygosity or epistasis, it takes more than one mutation to get to the harmful genotype. So you wouldn't see that drop off in fitness until later in the experiment. And it seems like around that kind of midpoint range, around like that 26th generation point, it's where you started hitting that tipping point where for whatever reason, the mutation accumulation started building up to where either you were hitting those homozygous genotypes or epistatic interactions that were causing issues. So here's the fun part here. My understanding, thank you, Jillian. My understanding level is that I know many of these words, so I'm just letting it flow over me. Here's the thing. This is not something that I've ever done in my life. And my degree is in molecular genetics, and I read a lot of these papers when I was in graduate school. Haven't since. And really, it was only in the first 2 years of graduate school. And then I was done with classes and I did my quals, and that was that, right? And then I was in my virus evolution bubble. So I haven't done this stuff in forever, and the specific techniques that we're talking about here are all new to me. So I can kind of make sense of it, but this is by no means something that I can just read and understand and explain, right? What I'm doing here is working through it with all of you and kind of developing my understanding by talking through it out loud, right? So let's see here, and then let's see. We've got. I want to see what this GERP. Where's that? Here we go. Genome evolutionary rate profiling scores, which evaluate functional constraint. So basically, you're looking at regions that are going to be experiencing more purifying selection versus regions that are less constrained, less purifying selection, right? And so you're finding mutations in regions that are more constrained in the kind of second half of the experiment, right? 2.4 versus 4%. And then the mutation spectra with transversions becoming going from 44 to 47. Okay, that's interesting. Yeah, so there's two types of point mutations. Transitions are when the size of the base remains the same. So it's purine to purine, so A to G or G to A, and then pyrimidine to pyrimidine, right? C to T or T to C. And then a transversion is purine to pyrimidine or pyrimidine to purine. So A to T is a transversion. And those are usually less frequent. Transitions tend to be more common than transversions because it doesn't mess with the structure of the double helix as much. And also it's just easier for the polymerase to grab something that's the same size that's wrong and kind of shove it in there. If it's a different size, that tends to not work, right? So that's just an interesting shift from 44 to 48%. I don't know why that would happen necessarily, but that's interesting. So large deletions identified in adjacent regions resulting in homozygous loss. Yeah, that's a big problem right there. What figure is this? Oh, that's a big figure. What figure is that? Oh, that was figure 4 up here. Okay, so this is just doing, okay. This is just the experimental procedure. Looking at blastocysts, seeing what happened, crossing the different lines, right? So BDF1 is the kind of control wild type, not wild type, but the control. And then G56, clonal generation 56. So you cross them and see what happens. Do we have to have experience in the software package to understand this paper? No, I don't know what software they used. And so, no, it's, we're just going through it kind of, we're not going to do the methods. Let's put it that way. We're doing the results right now, and we're not going to go through, I'll show you real fast. If you go all the way down here, after the discussion, we have down here, we get to the methods, and this is where it gets really intense with the specifics of what they did, because in general with the methods you need to provide enough detail that somebody can replicate your work if they want to. Between the methods and the supplementals, somebody should be able to do it. We're not going to do it to that level of detail here tonight. We're going to go through the results and see what's interesting. Okay, so we were up to this stage where we had a loss of a gene, which is important. That's definitely going to cause some fitness problems, and that is G57, across generations, too large to lead, but which generations? I want to know when that happened. I would have asked that. I would definitely have asked that if I was reviewing that. I want to know what generation that happened in. Considering the complete mutation spectrum, about 30 loss of function mutations. That's a big deal. But here's a really interesting thing. 30 loss of function and 50 missense mutations out of like 4,000-ish. It was 3,700 SNVs, and we're looking, and a bunch of indels. So it was like 40, yeah, so it was like 4,400, and they're saying, sorry, where am I here? I was up here. Here you go. Here we go, and it's like less than 100 mutations that probably had a fitness cost, although I bet the X chromosome lost may have as well. So in this context, the extensive background mutation load, the emergence of additional deleterious mutation in the final generation. Oh, so this was right at the end. The CSMD1 was right at the end, exceeded a tolerable threshold, leading to lethality or developmental impairment. So it looks like it is mutation accumulation that did it, right? It's out of like 4,000-ish mutations that accumulated over the course of this experiment. Like 80-ish had like clear fitness costs probably, plus there was the X chromosome deletion. And you put all that together, you basically cross that viability threshold when you get to generation 56 or 58, whatever it was, 58. Okay, so it seems like that we have an answer now to the question of what caused the decline in fitness. It was, in fact, mutation accumulation over the course of this experiment. Okay. And this is interesting. Strongly associated with accumulation after G23 to 25 rather than the effect of any single mutation. Yeah, so it's the mutation of it's the effect of lots of little mutations accumulating over time. So to compare mutation patterns in asexual versus sexual, we analyzed the sequence that's WGS's whole genome sequencing from 2 mouse lines, and then we get sibling mating. So these accumulated a bunch of mutations, not as many, but over 1,000. And with lots of homozygosity, as you would expect with a ton of inbreeding, right? So inbreeding is going to reduce the efficacy of recombination in terms of weeding out harmful mutations, because if it's siblings, right, as it was here, then they're going to be the same for a bunch of their genome, right? The coefficient of relatedness is 0.5, and after generations of inbreeding, that's going to go up, right? So you're going to see lots of homozygosity there. And then, okay, it's 1/3. So it is a 1/3 rate of mutation accumulation and lower indel frequency as well. And then they did whole genome sequencing again, produced by natural mating, right? So we've got our late stage clones plus wild type males and their grandchildren. And here we go, mutations in G55 were passed to both. In the supplement, you could see the specific ones. Translocation was also found. Interesting, because both affected chromosomes were inherited together. Now that's interesting. So I don't think they're going to go into this more, but if you have two chromosomes that swap a piece, and then during segregation, during meiosis, if you only inherit one of those translocated chromosomes, that's going to mess with it. You're going to end up with either monosomy or trisomy for a region. But if you inherit both together, they balance out. So interesting observation that was just happenstance there, but a cool thing. Okay, so let's get into the discussion. Now, before we do this, let's just talk for a second. And first of all, the discussion is what? It's a little bit there, and then this is all tables, so a little bit there, and then this page. So it's like, you put all that together, it's like about a page of discussion. So not a whole lot left for us to cover here. The point of a discussion is to interpret your results and contextualize them in kind of the broader field in which you're operating, right? So we want to say, okay, here's what we found. It means X, right? We saw X, or we saw X. We think it means Y. Here's the question we posed. Here's what we think the answer is, and here's why we think that's the answer. So now we're going to rehash the problem, right? It's unclear whether mammals could sustain clonal reproduction, 57 generations, not sorry, what did that say? Epigenetic abnormalities was not the problem, but mutations did, and then ultimately too much mutation accumulation killed the lineages. Okay. On average, each generation acquired 70. Oh, hang on. I see a comment here. Well, not that one. Sorry, Templar Warden. Did they test the mutation rate? Yeah, and it was like 60 something or 70 something per generation, I think they found. It was right up here. It was, I think it was up here where they talked about the mutations per generation. Yeah, oh, here we go. Yeah, it was 69.4 single nucleotide variants and 1.4 indels per generation. Yeah, so they did do that. And what they're concluding, it sounds like, is it's that accumulation of mutations is responsible for the fitness decline observed during the experiment. Oh, that's the translocation. That's really interesting. Huh. That's interesting. Okay, so here we are. We were here. So on average, each generation acquired 70 single nucleotide variants, 1.5 structural variants, large structural variants, comparable to those in somatic lineages, human germline. Yup. Okay, so the rate itself is not the problem, right? The rate is fine, but between doing cloning rather than sexual recombination means that there's a problem, right? So let's see. So two factors, long-term clonal propagation. Large structural variants, rarely seen in the germline because they'll often kill the germline, right? You have selection occurring in your germline where the germline lineages that have strongly deleterious or lethal mutations don't contribute to the next generation, right? Those sperm or those eggs don't get fertilized. So when you're cloning, you don't have that same problem. So those variants can persist, right? And then we got generation 25 during this period. Oh, here we go. Capable of regenerating clones, preferentially selected. So this is interesting. They're saying there could have been like a selection effect built in just to the experimental protocol, which I could see that, sure. But then once the mutations get to a certain point, the selection's not strong enough to deal with the accumulation of mutations through these clonal lineages. Advantages are not sustained. Yeah, it's the like I said. So this see, so after G25, The structural variations and loss of heterozygosity is your problem. So you increase homozygosity. And like I said earlier, remember I predicted one explanation for this time dependence could be that you have to increase the homozygosity, right? So the mutation occurs early and that doesn't cause a problem because it's recessive. But then if you get a second hit on the other copy of that gene that inactivates or damages that other copy, it could be the same mutation or a different one, but both copies experience a loss of function, now you got a problem, right? So that's what's going on here. Buildup of harmful variants outweighed adaptive effects. Positive selection effect results in the irreversible decline. Yes. And then the harmful mutation rate doubled around that time, which is consistent with what we've been talking about, either epistatic effects or homozygosity. Good. And then, oocyte, right, and development was affected. Okay. All right. So let's look at this next paragraph. The study was conducted 20 years. Variation is quality of culture medium. It was impossible to maintain complete right. So controlling this is going to be difficult, but you do have 2 of the participants did the full 20 years, which is awesome. No change in the birth rate of the cloned mice. You lost viability, but not birth rate. So that's one component of fitness that was not affected. Unlikely these factors. And then this is just a callback to the epigenetic part, right? So it's a histone deacetylase, so it removes the acetyl groups from those histone DNA structural DNA binding proteins for the structure of chromatin. And then let's see. Okay, yeah. Also reactivate endogenous retroviruses, which is bad. You don't want to do that, by the way. By the way, this is a fun little aside here. This is a really good evidence for junk DNA. The fact that you have regions like endogenous retroviruses that are inactive and then become activated through like mutations or something. And what many people and code and creationists will say is that, oh, by the way, that region becoming mutating and then causing a problem is evidence that it's functional. No, it's not, because when it's locked down, it's not causing a problem. And then when you disrupt that and it becomes active, now it causes a problem. That means the lockdown version was not doing anything, right? That's the opposite of function. So just a little aside there that this is kind of brushing up against there. Okay, let's see. Unlikely TSA, yeah. Okay, so it's not that was causing the problem. Late generation clones survive normally healthy, normal lifespan. Yeah, placenta size increased. Okay, so they're going through like, we check this, we check this, we check that. What's the actual problem? Samples, so if it have a critical mutation that cause lethality. Okay, here we go. Preservation of reproductive capacity, 20 years. All examined clone mice retain fertility. Sexual system function is not required for the individual clones. It's improbable that only cloned embryos with normal reproductive systems were selected. Instead, they imply that the sexual reproductive system is robust against mutational damage. That's pretty cool. And would make sense, right? You would expect some degree of redundancy there, because if you have redundancy and you are obligately sexually reproducing, then you're going to be able to reproduce even if you experience mutations. If you lack that robustness, right? The phrase is mutational robustness. It's how many mutations can you tolerate, essentially. If you lack that robustness, then you're more likely to be unable to reproduce. So you see selection for robustness specifically in regions of the genome that are associated with sexual reproduction, right? That makes a lot of sense that you would have selection for that phenotype, robustness being the phenotype there. This is something you can even see in viruses. Mutational robustness is a thing in viruses for basically the opposite reason, that viruses are asexual, reproduce really, really, really fast, and have higher mutation rates. So you need to be able to tolerate all those mutations. So you, in some viruses, can document selection for mutational robustness. It's a cool thing, and you see that if you calculate a distribution of fitness in terms of like doubling time, for example, the more robust populations have a flatter distribution, right? So as the x-axis is your mutations and the y-axis is your fitness, the flatter that distribution is, that indicates that you are robust against mutations, and that's a dynamic you see in viruses as well. Okay, so throughout the study, oocytes collected from young individuals. Yeah, right. We want to minimize the age-related stuff. Okay. The necessity of sexual reproduction explained by the Red Queen hypothesis, maintain genetic diversity, and enable species to persist under changing environmental conditions. Right, that is one hypothesis, and there is evidence for that. For example, in populations where you have you could reproduce sexually or asexually, you see that sexual reproduction is correlated with parasite load. So the more you're competing with parasites, the more recombining you want to be able to do. So there is evidence for the Red Queen hypothesis there. However, that's obviously not going on here, right, in this experiment. So that can't explain what's happening here, and specifically can't explain the recovery when you take those late stage clones and cross them with the wild type, right, or the control line rather. So here we go, sexually producing, generating diversity, even without recombination. Here we go. Right, so they're generating a lot of mutations. So you're generating the diversity without recombination. So the problem isn't, I see what the authors are saying here. Got it. So what they're saying is, under Red Queen, the variation is the point, right? In and of itself, generating the variation is the point. What they're saying is no, because the asexual line has even more variation, right? So if variation equals good, like that's why this system exists, well, you can't say that because here you're getting even more variation. So then you, instead of looking outwards and saying the variation allows you to deal with environmental change, you have to look inwards and say, okay, what's happening in the genome that recombination facilitates that clonal reproduction doesn't, right? You're still getting variation, but what is it about the variation from recombination specifically? And I think they're going to go into this here. Bingo, Muller's ratchet theory. That's the other hypothesis. Love how we're just hitting all the hits, right? We're playing all the hits here. So Muller's ratchet is the idea that in asexual populations, you have an inevitable accumulation of harmful mutations, even if good mutations happen too, because there's no way to decouple them from each other. So you get bad, 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 ooh, good, bad, bad, good, bad, 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 good. Well, you're selecting for the good, you're dragging all the bad along with it. In sexual populations, and that's Muller's ratchet. In sexual populations, that doesn't happen because you can have recombination and decouple good from bad, right? Or I guess I did the other way. You can decouple those mutations, and that increases your fitness, right? It allows you to select for the good things without having to bring along the bad things with you, right? And looking at how their specific findings and how they describe them, right? You have an increase for half about to generation 26, and then from there to 58, a decline. And what that implies, I think, is they described a little bit of the selection that could occur in there. I think what's basically happening is, in that Muller's ratchet paradigm, right, the good mutations are kind of driving the car. They're in the driver's seat for the first half, but then the accumulation of all those bad you bring along for the ride, they grab the wheel in the second half and cause the fitness decline. Yeah, I think that's a reasonable explanation of what's going on here, right? So deleterious mutations accumulate, mutational meltdown, and extinction. Yes. And so mutational meltdown in this context here is not just the mutation accumulation, it's also the homozygosity, right? Because it's new variants, but then you end up having two broken copies of a gene that you need. And that's a problem, right? So our study provides the first empirical demonstration that mutational meltdown as predicted by Muller's ratchet can indeed occur in mammals as long as they are clones. In comparison, wild-type mice maintained through natural breeding showed no fertility decline, emphasizing the mutational load of clonal reproduction exceeds a threshold compatible with long-term mammalian survival. At the same time, the observation that recloned mice maintain healthy insight into the extent of mutation tolerance. There you go. So we can wrap this up here and basically say, look, we have in clonal mice, we have mutations accumulating, and that is ultimately going to cause extinction of this population. And then when you outcross them, right, with the non-clonal mice, and you recombine, the non-mutated genomes back in, you recover, not all, but some of that viability and show that you can arrest that decline in the fitness of that population, which is really cool. So let's see how they wrap this up. Dolly the sheep, can we clone things? Can we, save endangered species? Yet these findings demonstrate the practical application of cloning requires a deeper appreciation of constraints, further research, reaffirm the inevitability of sexual reproduction as indispensable long-term survival of mammalian species. Yeah, basically what this is saying is the ability to decouple wild-type genotypes or beneficial mutations from harmful mutations leads to perfectly healthy populations, perfectly sustainable mutation load, mutation rates, mutation accumulation, all that stuff, right? Whereas if you clone the populations, you are removing one of the ingredients from that mixture, the recombination, the decoupling, and then Muller's ratchet takes over, and now you are unable to clear those mutations, and you build up mutations until you hit kind of that critical point, and then no longer is that population viable. Okay. That's pretty darn cool. That's really cool. So what we can do now is anybody have any questions? We could just hang for a few minutes and I'll see if anybody has any questions, anything that you didn't understand, anything you thought I missed. Feel free to put it in the chat and I will I'll see if I can clarify anything and then we can go from there. So this is an interesting one, right? Would that fall under error catastrophe? I would have to look at the math because error catastrophe describes a very specific situation, in terms of the population genetics of what's happening, but I will say that it could, right? So I was making the joke genetic entropy does happen in this situation, right? That's what creationists are going to say. They're going to be like, look, it's an experimental validation of genetic entropy. And in the same way where they'll say, they'll model a waiting time problem for human evolution using haploid asexual populations. This is not a realistic mammalian population, right? It's literally clones. And mammals don't reproduce clonally. So if you need to break mammalian reproduction to the point where it's clonal in order to get genetic entropy to happen, that doesn't say much for genetic entropy happening in natural populations, right? Yeah, interesting. This is a good one. From Ration Extrema 3776. This has interesting implications for species that utilize parthenogenesis and self-fertilization, and why males still persist despite that strategy. Yeah, you have to do the recombination, right? You can't get away with it otherwise. Does this help the cheetah population? Cheetahs need outcrossing. Cheetahs are super inbred due to a bottleneck that happened it was a long time ago. It might have been tens of thousands of years ago, I think, as far back as 70,000 years. So yeah, the cloning is not going to help them. They need less cloning. They need more variation, less heterozygosity. You just disproved evolution because genetic similarities don't mean anything anymore if you can't use the mutation rate. I don't know what that means or if it's serious or parody. Gypped Gamer 9723. Tell me if that's serious or a parody, because I'm not sure what you're saying. Okay. How much different details would be different with non-mammalian? Yeah, it would be interesting to see, like, do this with reptiles or do it with frogs, right? Do it with amphibians. The shorter generation time too. That'd be really cool. Okay. Yeah, this is I love how everyone's like, and here's the next thing you should do, researchers. Yeah, let's look at parthenogenesis in other populations, especially like vertebrates that do parthenogenesis. Yeah, I think that's really cool. That would be a fun thing to check out. I will say that there are animals that reproduce sexually and asexually, and you do see a correlation with parasites, more sexual when there's the parasites. Okay. So I think that's where we can leave it, folks. So thanks for joining me as I went through this paper. I thought that was really cool. I hope you all enjoyed it. Before you go, please do me a solid and hit that button down there with the thumbs up. Or if you really, really, really hate biology, you can also hit the thumbs down. I don't know why you would be here if that was the case, but thanks anyway. And also remember to subscribe and tell your friends to subscribe and whatnot. And in terms of what I have coming up, I plan to record something for next week. It'll probably be on the shorter side just because time of the semester, and I have a few things in the works that I'm like pretty close to ready to record. It might be just one of those real quick, here's how you beat this creationist argument things. And then I'm also in the process of setting a date with some people for the first of the creationist boot camp scrimmage type streams that we're going to do. We'll do that soonish, soon as I get everyone together and we lock down dates. So that's coming up soon. And I think that's where we can leave it. So thank you, everybody. I appreciate you all being here. Keep an eye out for what's coming next time, and have a good night. Don't get fooled. Bye, everybody.